So now we understand the role of fluctuations in small systems in a rather generic way. We turn now to an attempt to understand the fluctuations in detail by modeling nanosystems with molecular dynamics. One of the great promises of science at the nanoscale is that the number of atoms involved is small enough that it is possible, realistically, to model these systems on large computers, subject to some constraints that we'll talk about in a minute. If you remember in Chapter 2, we wrote down an example of a model interaction potential between two atoms as this Lennard-Jones 612 potential. So with these parameters known, one can then write down all the interactions between an assembly of atoms and work out their mechanics from Newton's second law. The potentials don't have to be as complicated as this even, as we have discussed at several points during the current series of lectures. Most interaction potentials around their minima can be described as harmonic potentials, so that the energy looks like this, from which one can then ascribe a force constant between the ith and the jth atom as the second derivative of the potential with respect to displacement. So armed with force constants and particle masses, one can then set up some model of the system, set it into motion with random velocities given by the equipartition theorem as shown on this slide, and then see what happens to the system. The overall process is limited not just by the number of atoms, but by the time steps required to make the computation. So for example, if vibrations are on the picosecond or sub-picosecond timescale, then the time steps will have to correspond to a very small fraction of this, perhaps 10 to the minus 13, 14 or 15 seconds. So there will have to be many calculations per time step, and given the speed even of supercomputers, this limits the range that can be reached by molecular dynamics. Now, let me show you, before we talk about the limitations of molecular dynamics, the output of an MD simulation. They're beautifully seductive things because they look so pretty, and one tends to believe pretty movies, so a word of caution. But this is an illustration of some work in our lab where we built the system that you see here. It's a piece of single-stranded DNA tethered to a surface, and the red ring is a cyclic sugar molecule that is actually pulled over the DNA by an atomic force microscope probe, and we used molecular dynamics simulations to try answer the question, could we tell which base was sliding through the sugar ring by the forces that were generated. We also actually built this system with a lot of chemistry, but that's another story. So what about the limitations of molecular dynamics? Well, it turns out that for biology, one really does need to go to rather long timescales. So you can think of these timescales in a number of ways. For example, for the long-range fluctuations of a protein embedded in a viscous solvent, we show in Appendix D that the damping time for motion of this particle is given by the expression on this slide, and that can turn out indeed to be all the way from microseconds through to milliseconds. Then another way of looking at this is that many processes have transition states that are quite high in energy, and so the intrinsic rate of sampling this second potential well here is very small. And so easily in biology, these timescales can be on the order of milliseconds. Timescales like this are really beyond the reach of conventional molecular dynamics. Another problem presented to molecular dynamics is that many true bonding interactions, like hybridization, are just not described very well by a model two-body potential. So there are improvements to molecular dynamics, and they allow one to extend the effective time of the calculation out to very long times, so that a true thermodynamic sampling of the states of the system can be made, rather than just a sampling of the states at short times. So typically a few nanoseconds is the limitations of a large simulation. So supposing the energy trajectory that a particle might go through is known. So if one can figure out the lowest pathway through some energy landscape, then what can be done is one can look at this trajectory by breaking it up into small regions like this, running a molecular dynamics simulation within the small region, and asking about the rate at which the particle leaves the region in the forward and reverse direction, and then setting up a set of master equations that couple these motions that were done by sampling a small region of the system and put them together to generate an overall rate of passage for the particle through the system. It's a technique called milestoning, and what it does then is to give one the parameters that are needed, the diffusion constant and the transition state height, for modeling the system with a Kramer's model.